what I'm going to talk about today is the aspect of skin sampling, which when I agreed to do it, I thought would be quite easy, but actually it was quite a complex subject to take on. So when you're thinking about sampling skin for preclinical studies, then you have to consider who you're sampling from, where on the body you're sampling, what you might be sampling. There's all sorts of things now that can be skin and then analysed, how you're going to do it, whether it's you're actually taking some material off or whether you're using instruments. And then what does it all mean when you've got your data? How do you pull it all together? So in relation to who, then, well, you know, we live in a very diverse market, as you've heard from Rachel. So when you're designing a study and you want to look at, you know, sort of the stratum corneum skin microbiome on males, you know, you have to decide whether you're going to pick just any old males or whether you're going to look at some of the geographical variation or age. Maybe look at things like issues to do with diversity, somebody transitioning gender. Are you looking at people in the city, or you're looking at people who live outdoors? You know, is pollution something that is an issue? So when you're really considering all these things, then designing your study based on the consumer population of interest is really important. Skin is hugely variable across all body sites, and this is a diagram for the skin microbiome that's been published. There's many versions of it published, and you can see that sampling from different areas gives you different types of information. And I think, you know, what we would say is that you can't really substitute one body site for another. There's no point in looking at the forearm if you're interested in the axilla. And, you know, if you're interested in the scalp, there's no point in looking at the abdomen. So I think some of the issues to do with sampling relate to the fact that you might need to take a sample from somewhere where it's quite difficult. Also, you've got different types of sampling methods. You use different instruments to do it. And then there are several other factors that might complicate this, to do with whether you do it in a clinic or you're doing it in somebody's, they're doing it themselves in their home, etc. So when we talk what, then this is a kind of a list. It's probably not an exhaustive one. So you might be sampling corneocytes, and they may be immature or mature. And you can use this diagram here shows you corneocytes from the forearm and the cheek. So you can see that even now with a marker look at the desmogleins shows that there's already a difference from different body sites. You might be interested in different microbial species. There's sweat and pH. It could be something of interest. Are you looking at sebum, mucous fluids, pollutants, metabolites, proteins, lipids, and then nucleotides? And what we know is that there is a vast amount of variation in each of these body sites with each of these things that you can actually sample. So it's a very complex area to be working in. So when we think about how, I've just illustrated a few of the kind of major methods. So a skin wash, so it could be swabbing, which would be with it's not very clear here, but with the sort of thing that we're all used to sticking down the back of our throats with COVID, you know, a sort of a swab with a bit of cotton on the end, or a buffer wash where you put on a little sealed ring and you put in some buffer, and then you would actually just sort of agitate the surface of the skin and then take off the fluid and analyze that. And, you know, you have to have really clear protocols for doing this, so that you need to know which method is the most important for which body site, for which analyte, etc. Another very, very commonly used sampling method is tape strip, often using D-squames. And this is a D-squame here. You can see it's a small round sellotape adhesive with a little area that you can grasp with your forceps. And when you sample that, you can take off stratum corneum, you can take off peptides, cytokines, inflammatory mediators, and microbiome components. And in this particular study, the effect of tape stripping was looked at. So after the skin had been sampled with a tape strip several times, you're looking using optical coherence tomography imaging, and you can see there's a difference before and after in terms of the surface topography to show that you're actually taking some materials away. And then you can use extract the tape with various solutions, and you can use it for full-on proteomics, or you can look for individual components using something like an ELISA, which uses a specific antibody. And some examples of this were Anna Burke-Mary. She did a PhD looking at the differences between atopic dermatitis and psoriasis, and she was able to take samples of the stratum corneum, do full proteomics, and find markers that separated these two conditions, which for the clinician is quite useful because often these conditions can present quite similarly. I would say it's not really suitable for moist skin sites, obviously, because the adhesive wouldn't stick very well. But that's maybe a gap where we need to come up with a nice, simple method for sampling for areas of sweat or areas of mucosa. And another one is where you absorb your analytes of interest into a material, and there's various things that you can do. So you can use, for instance, if you're sampling sweat, and you can measure electrolytes and metabolites, and then you can use different types of sensors to do this. Biosensors, which detect a particular material, or ampermetric, which detects change in the current, and then ion selective again, which could detect, say, just the chloride ion. And this scheme here comes from a paper that was published a few years ago, where they developed a little patch with a microfluidics arrangement, and on induction of sweating, you end up with a change in the colors, the colorimetric presentation of different materials that are being examined and analyzed for. And then you can look for the different materials like lactate, chloride, change in pH, and glucose. So these sorts of things are becoming ever more useful for complex analytes. And certainly anybody who's interested in using sweat as a vehicle for measuring something that's physiological, like, say, glucose for diabetes monitoring, these types of devices are going to become more the norm. Then we've got sebum, and we've got the good old Sebutape, or even a Rizla paper, which is, just a cigarette paper. Sebutapes are manufactured and have a sebum, a lipid absorbent layer, and when you apply it, you can see no black. And as the amount of sebum collects on the Sebutape, you get more and more areas, and they're black because the sebum changes the optical properties of the tape. And when you look at it using your special instruments, then you can actually quantify the area of sebum, and that gives you a gravimetric, the equivalent of a gravimetric amount. Just a little fact here. Did you know we secrete between 500 and 700 mils of sweat, i.e. hypertonic fluid, every day? So it's very important to keep drinking more. And then the last area is how you sample mucosa. And I think this is one where I found it more difficult to find information, and again may represent a sort of an opportunity. So the female genital tract, for instance, has a great complexity. It has different layers, different levels, upper, medium, and low. And it has many, many different materials in there, whether they're proteins, carbohydrates, or whether you're measuring the pH, or whether you're collecting samples of the microbiome. So typically vaginal swabs, which are again very similar to the kind of COVID swabs that we use, and something called a cytobrush, which is like a little tiny pipe cleaner. And they are typically used for collecting upper vaginal swabs. more normally for detecting things like infections. However, there doesn't seem to be anything that's good for collecting the normal mucosa, perhaps in the mid and lower part of the vagina. And for people who are interested in sampling that for a microbiome analysis or looking at the influence of, say, estrogen treatments, that's something that could be an opportunity. So when we've completed our collection, we have to do analysis. And there are several ways to do it, but I've highlighted the three that I think are most common. So there's proteomics, and proteomics analyzes the proteins that you've collected in the sample, and it does using mass spectrometry. And basically what it does is the whole protein solution is converted into peptides. Those peptides are separated based on their mass, and then the accurate mass of a peptide is calculated from the known, the whole proteome is known now using databases, and you can identify a peptide by its mass, and that enables you to identify proteins based on their fragments. And then lipidomics similarly collects information on the lipids in a sample, and it uses methods to detect volatile lipids like gas chromatography, but also non-volatile and covalently bound lipids, maybe using liquid chromatography. And then you can separate these out and determine the proportions using standard benchmark materials. And then more recently, we've come into the variety of microbiota in the sample, and there are essentially many ways to do this, but the most common is to do called 16S sequencing, and it is 16S is a part of the microbiome, which is part of the microorganism, which is unique to microorganisms and isn't found in humans. So you can separate the two that way. And then when you've done all that, you get masses of data. So you need to have good bioinformatics to organize that data, generate lists of the most important materials that you've identified in your samples. And then now that's linked to machine learning, and machine learning basically looks for patterns in data. And you can train an algorithm by giving it samples of data which characterize what you're looking for quite carefully, and then you put in a naive sample and it can actually tell you what the patterns look like. And all of that leads to meaning in terms of, what does it all mean, new knowledge, what you might want to learn from a disease or from the normal physiological state. So a few case studies. So for proteomics, for instance, using these frames, a group have looked at female facial skin, looking at the photo versus the photo protected. So that's looking at the cheek versus the bit of skin behind the ear, which is generally photo protected. And this is the result of the analysis they get. They can show the proteins that are involved in corneodesmolysis, which is involved in desquamation, by filaggrin analysis, which is important for the production of the skin moisturizing factors, and then the cornified envelope maturation. So they're able to look at the differences and show which are up and down regulated. And they found that a particular protein, which is upstream of cornified envelope maturation, was identified as a new target for photo-damaged skin as its levels were lowered. But also they concluded that there was poor crosstalk between the keratinocyte endoplasmic reticulum, which is in the cell, the proteasome network, and autophagy machinery that leads to impaired keratinocyte proteostasis. So in the cell, all of these processes are used for giving you normal healthy skin, and if you find that any of those are affected, when you compare your photo age with your photo protected, then you can show information that tells you about the mechanisms behind skin aging, and that might lead you to some novel ways to intervene. In this case study, this is Lipidomics. This was a study with dark-skinned adolescents with and without acne, looking at the forehead and the cheek, and they used the Risler papers for this. So this is a different type of presentation showing the different lipids. You can't see all the, probably can't see the detail here, but the main thing is to show is that the control and the acne, which is the blue and the red, show differences between obviously the control and the acne, and also differences between the cheek and the forehead. And this type of heat map is very commonly used to present large data sets to show pattern differences between, say, two body sites or two different conditions. And the sorts of things that were being analysed were triglycerides, wax esters, and those were done using high performance thin layer chromatography. And then things like free fatty acids, fatty alcohols, squalene, cholesterol using gas chromatography. And in the case of a microbiome, this is a study that was done on the scalp in India. And what they did was they used a wet swab in this case. So they had a buffer dipped wet cotton wool swab, and that was wiped with a fairly standard protocol across the scalp. And they compared dandruff and non-dandruff. And they came out with the conclusion that P. acnes and Staph epidermidis are significant bacterial species on the scalp. We often thought in dandruff that it was all about the fungus, but actually the bacteria are very important. They did find that the fungal microbiome was enriched in pathways. That's looking at the physiology, implicated in cell host adhesion in the dandruff scalp, which is quite interesting. Because obviously, if you've got a microorganism that's living in a sort of skin surface, that adhesion is important for sustaining the colony of the microbiome. And then they found some Malassezia species which were as yet unclassified that were strongly linked to dandruff. And they concluded that the bacterial microbiome provides the scalp with essential nutrients to maintain a healthy state. So this is like more of a positive finding from the presence of the microorganisms. So we can also sample the skin using instruments, and I've just put here the confocal Raman. I'm by no means an expert in it, but it's a non-invasive way to see into the skin and also to detect chemical species. Whether they're endogenous metabolites, water, or drugs that have been added topically. And in this particular condition here, you've got different body sites that have been looked at, and you get a profile of different body sites, and there are slight differences. And those Raman spectra can be used to compare different body sites. And then in this example underneath, a Raman profile was used and was able to detect a benign tumor signature versus normal skin, and they're able to show that how the tumor and the normal varied. So these kind of non-invasive instrumental methods are also useful and complementary to the preclinical ones where you're sampling. It's really important to think about how these things all work together. So, you might think of sebum, microbes, and pollutants. What's the relationship? So if you're measuring things on the skin, you might want to think about how they all interact. An example perhaps is the relationship between the microbiome and the skin's production of antimicrobial peptides. So RNase7 is secreted by keratinocytes, and it's important for microbial skin defense in both health and disease, and therefore can be used as a biomarker, and its expression is triggered by the interaction between the microbiome and the skin. And in another example, I found the pollution and the facial microbiome shown to have a strong relationship. And the conclusion from the study was that skin microbiota is first to respond to air pollution, and that measurable skin benefits come from maintaining a stable, strong skin microbiota with good connectivity between each other and the skin. So measuring one thing probably isn't enough, and if you're doing preclinical studies, I think that it's important to do quite a few different analytes. So just to summarize, sampling human skin is complex. Be clear about the study question to address and design an appropriate study. Consider which surface materials to measure and how best to do this. Think about the interrelationships that are important for skin health in your study design, because there are going to be several of them. You can't generally use one skin site as a substitute for another. And innovation in the skin sampling kits is needed, especially for in-home or remote sampling. And like I said, possibly for mucous membranes would be an area for development. And I'd just add the COVID-19 effect. We're all scientists now, we've all done sampling. So I think it's a very good time to get this type of activity out into studies and into the general market. So that's the end. Thank you very much for your time, and I'll be happy to take any questions.